will remarkably make its way past the blood-brain barrier and target motor neurons within the spinal cord. In mouse studies, we have 60 to 70% of the motor neurons throughout the entire spinal cord that are expressing the gene. Now, there's a window of opportunity in the mouse. We've been funded to to answer a question: is there a window of opportunity in the non-human primate? That is, if I deliver a virus through the vasculature in a non-human primate, will we still target motor neurons in a newborn monkey, in a 3-year-old monkey? The answer is, even in a 3-year-old monkey, we were targeting motor neurons fairly efficiently. So we have expanded the window of opportunity in a mouse, which appears to be a shorter time frame, and expanded that in the non-human primate. And we have a few more studies to do, but we're quite encouraged on the aspects that we can target motor neurons via a vascular route of delivery in a 3-year-old non-human primate.
Okay. And another question from Ashley. What does he believe the first clinical trial will look like, such as age of child, number of children enrolled, only one injection, which we've answered, as opposed to many, and at what point, if they see great results, would the FDA open the clinic up to more children, or would it only be open to phase 2 of trial?
So once again, these are excellent questions, Ashley. Some of them I can speculate upon. So one portion of the investigational new drug application is the actual design of the human clinical trial. That is, what would a phase 1, what would a phase 1/phase 2 trial include? It appears, and basically nothing is set in stone until one has the meetings with the Food and Drug Administration, which again we're trying to plan for January, for the first quarter of 2011 to be speaking to the FDA. It's there where we will propose the human clinical trial. Now, we have a clinical trial group that includes physicians, neurologists, and collective groups of basic researchers and clinicians who are thinking about the design of the human clinical trials. Speculating once again, but our feelings are to the first clinical trial would be in a type 1 patient, newly diagnosed.
Okay. And I'm sorry if some of these questions get to be repetitive. I just wanted to ask every single question that the families have relate to us. So, Mary...
Absolutely. I think I need to answer on Ashley's questions. Would it be opened up in phase 2 of trials?
Right.
So first off, the first trial is looking for safety, hints of efficacy. It would be a small cohort. Likely it would be a small cohort of patients, type 1 patients, newly diagnosed, where one is collecting the safety data, where one is collecting hints of efficacy. If successful and proven safe, one would then, in hints of efficacy, one then starts expanding upon those trials for larger numbers of patients, and one then can think about the opportunities for targeting other types of SMA, that is type 2s, type 3s, or later stage type 1s.
Okay. Okay, so Mary says, thanks for all you do to help find a cure for SMA. Thank you as well to Dr. Kaspar for his willingness to help out families in such a personal way. My question is similar to Ashley's. When will a child with type 1 SMA be too old or large, length of weight, to benefit from his therapy? For the older, larger children, what does he see as the best long-term management for increasing strength?
So Mary, thanks very much for your questions. Here we have the questions again. What is the window in an SMA child or an SMA patient that one will have efficacy? The honest answer to that question, if one puts SMN back into a patient, one doesn't know the window of opportunity for where SMN will work, when it won't work. Our hopes are, the hope is that if you put SMN back and the motor neuron still exists in the patient and the motor neuron has the proper connection still and it's sitting dormant, that will be able to awaken that motor neuron by the SMN, and that motor neuron will restore some or all of its functions and restore normal physiology. Uh, there really are no animal models that allow us to test and to answer that question at this time. There's a number of groups who are working on different animal models to try to, including our own group, to try to help answer some of these challenging questions. Some of these questions are likely going to be answered in human clinical trials in the experimentation stages.
Okay. Ashley, once again, she has a couple more good questions. She wants to know if you will apply for compassionate use of gene therapy with the FDA.
There's certainly discussions that need to be had with the Food and Drug Administration, including compassionate use. Again, this is an experimental gene therapeutic. So while we do studies in our laboratory and preclinical studies that have demonstrated significant results that have ourselves, clinicians, and everyone in the field excited. And I do want to make another note. There's been several gene therapy studies that have been published that have confirmed that if you put SMN back via the same virus that we're utilizing, adeno-associated virus, that all three of these groups have found significant extensions of lifespan in the animal. So I think this adds to a confidence that gene delivery, if one gets it to the motor neuron, will have an effect in the mouse model of the disease. That gets us all excited. Then we need to test this experimental therapeutic to gain confidence of truly, is this going to be safe for humans? This is the paramount concern for moving into human clinical trials, and it's the paramount concern in what's being tested in a phase 1 clinical trial. So we'll be looking at all of these things very carefully and really evaluating the safety in preclinical studies and then formal toxicology and safety studies that are agreed upon by the Food and Drug Administration. Once one moves into phase 1 clinical trials, one can then start seeing signs of efficacy, and our hopes are for efficacy, and then to apply for compassionate use status. It's too early to tell right now, but certainly it would be considered. The other thing that I want to mention is that we also are in the process of thinking and applying for Orphan Drug Act, and those are there's a number of different things that or orphan drug status that would position the development of this and certainly involve the Food and Drug Administration in those aspects for SMA.
Okay, Ashley also asks, what does he believe gene therapy will help the most? With respiratory strength, with movement, with everything? Or how does he believe it will help, and to what extent?
So here, those are good questions. Again, Ashley, and again, it's an experimental therapeutic that is being tested in human patients, and there will be experimental endpoints that we have to define and measure to determine whether we're seeing any signs of efficacy in the initial studies. So those questions are really being developed as part of our investigational new drug application, where the neurologist and the individuals who will be seen, who will be treating the patients, who see the patients, will really be developing the endpoint criteria. That is, one, safety, and then the second endpoint criteria are the hints and signs of efficacy in a small cohort of patients. And certainly from our mouse studies, we had a lot of correction. That is electrophysiological correction as well as motor functional correction. And then finally, lifespan extension, significant lifespan extension in the mouse model. Certainly similar sorts of testing would be done in clinical trials.
Okay, Ashley wants to know, is your pre-IND meeting scheduled yet? And if not, when?
So we have not scheduled the the pre-IND meeting. The reason that we have not scheduled this meeting yet is twofold. One is we're preparing or we're in the process right now of doing those safety studies in both mouse and non-human primates for us to get a nice handle on the safety. To date, all of our safety studies in mice have proven that our SMN gene delivery is safe and well tolerated with no side effects. We're moving into non-human primates for testing there. That gives us confidence to present that data to the Food and Drug Administration to help guide in the formal toxicology and biodistribution studies that we would propose in the pre-IND. We want that information at hand so that we can utilize that to help guide what we would propose for a formal biodistribution and a formal toxicology study to move forward for SMA clinical trials. We're hoping to have the second is the pre-IND materials are being planned right now, and it's significant planning from how we would make the virus for human clinical trials, how we test the virus for human clinical trials, what sorts of safety studies that would go on, what would the first clinical trial look like. All of this material turns into several inches of papers that would be submitted to the Food and Drug Administration, and really is collectively pulling together biologists and clinicians on designing these. And we're in the process of doing this and making significant progress, as well as we're developing that safety data, mouse and non-human primates, and moving along quite fine. Again, our hopes are to have that meeting, or the paperwork submitted, and having the meeting in the first quarter of 2011.
Okay, so just to clarify, the initial studies you had done were to prove efficacy, how effective this was in the SMA animal model and in the primates, and now you're doing the safety study showing how safe this therapy is in those animals, correct?
That is correct. We've used a mouse model for efficacy on motor function, so SMA mice. Can we correct the disease in the mouse? There is no model of SMA, or monkeys, non-human primates, do not develop SMA. So we're utilizing normal non-human primates for two reasons. One is, in a non-human primate, something that on size and something that is closer to the human, can we target motor neurons? What is the window for targeting motor neurons? The second aspect is, in a species that's more similar to humans, is our drug, is our gene therapy safe and well tolerated? And that is where we're taking normal non-human primates, injecting vascularly the gene therapy encoding the SMN, and then doing a number of tests in those non-human primates, a small study, to give us confidence on the safety there.
Okay. Ashley asked another good question. Do you believe that children living today with SMA will benefit from this gene therapy, or do you believe it will be more for children born in the future with SMA?
So this is an excellent question, and I think one that is questioned not only for gene therapy but for drug therapies, oligonucleotide therapies, and others. That is, can one go in to a later stage patient, increase SMN levels via any approach, gene therapy, drugs, oligonucleotides? Will that have a response in the